Hi everyone, my name is Liz Renner and I'm a PhD candidate in Keith Ghetto's Fish Ecology Lab at Kansas State University. And today I'll be sharing with you some of my dissertation research on the impact of gizzard shad on water quality in Kansas small impoundments. So cyanobacterial blooms present a growing public health issue in lakes and reservoirs across the United States. Evidence is mounting that harmful algae blooms are increasing in both frequency and severity as a result of climate change. Drought conditions can reduce lake levels, concentrating nutrient-rich agricultural runoff and potentially triggering algae blooms. In addition, torrential rainfall and flood events can also trigger sudden influxes of agricultural nutrients that can trigger blooms. Under certain conditions, cyanobacteria can release neurotoxins, hepatotoxins, and dermatoxins, posing threats to the health of people, pets, and livestock that come into contact with the water. According to the Kansas Department of Health and Environment, there were 244 confirmed harmful algae blooms in 95 different public reservoirs managed for drinking water and recreation between 2010 and 2020 in Kansas. Recently, there have been questions over the ability of benthic feeding fishes such as gizzard shad and common carp to resuspend nutrients off of the bottom of these reservoirs, potentially fueling cyanobacterial blooms. One of these benthic feeding fishes is the gizzard shad, Dorosoma cepedianum, and they're the subject of my dissertation research. Gizzard shad are freshwater members of the herring family, Clupeidae, and they have a very unique life history that makes them particularly capable of dominating reservoir ecosystems. Gizzard shad reproduce quickly, experience rapid growth, and are facultatively omnivorous, meaning that they can switch from feeding on zooplankton to benthic detritus when competition is high. A number of previous studies have documented positive relationships between gizzard shad abundance and ecosystem productivity, quantified as chlorophyll A concentrations, along with total nitrogen and total phosphorus concentrations. Higher shad abundances have also been associated with reduced zooplankton biomass and size structure. High shad abundance also tends to be negatively associated with the abundance of sport fishes, like bluegill or largemouth bass, which compete directly with shad as juveniles for limited zooplankton prey. Finally, a study by Shad et al. Analyzing gizzard shad excretion suggested that the low N to P ratio at which gizzard shad excrete could alter phytoplankton community composition in favor of the cyanobacteria. Now, it's thought that gizzard shad play a key role in providing nutrient subsidies to phytoplankton via excretion of sediment-derived nutrients. They then excrete and resuspend that nitrogen and phosphorus back into the water column, providing a nutrient subsidy to stimulate phytoplankton growth. This can become a positive feedback loop, and gizzard shad can facilitate the eutrophication of small impoundments. We know that gizzard shad can influence small impoundment limnology, but we don't fully understand how limnological parameters co-vary with shad abundances. So to summarize, gizzard shad are ubiquitous, and cyanobacterial blooms are becoming more prevalent, and there's growing concern about their management costs. However, we still don't fully understand the role of gizzard shad within food webs, and to our knowledge, no one has ever directly studied their effects on water quality via replicated lake manipulation experiments. Therefore, the overarching study objective for my dissertation project was to evaluate patterns in nutrients, primary productivity, and zooplankton communities across a gradient of gizzard shad abundances. So we asked, are there associations between gizzard shad abundance and the limnological characteristics of Kansas small impoundments? At higher shad abundances, we anticipated greater nitrogen and phosphorus concentrations, higher algal densities and phytoplankton communities dominated by cyanobacteria, lower water clarity, and zooplankton densities, as well as zooplankton communities dominated by smaller bodied rotifers and copepods instead of dominated by cladocerans. For this experiment, we collaborated with the Kansas Department of Wildlife and Parks to sample 12 small impoundments managed for recreational fishing. The impoundments all had similar fish communities, but varied in gizzard shad densities. Our first step in this project was to quantify gizzard shad abundances to determine whether or not a shad abundance gradient actually existed in these impoundments. We did this using a combination of two techniques: standardized fall gillnet surveys conducted by the Kansas Department of Wildlife and Parks, and daytime shoreline electrofishing. And we saw some concordance between the two sampling techniques. Ultimately, we settled on using our electrofishing survey data for our models because it was collected on the same temporal scale as our limnology sampling. So for our data collection for this project, we went out and collected samples once from each impoundment every June, August, and October from 2017 through 2020. We sampled 3 stations per impoundment: up lake, mid lake, and down lake in the deepest area near the dams. At each station, we measured Secchi depth, collected surface water grab samples for total nitrogen and total phosphorus. We did repeated vertical and horizontal zooplankton tows for zooplankton and collected pelagic algae samples. For our experimental design, we had a regression design because we were interested in relationships between shad abundances and specific dependent variables across environmental gradients. We fit generalized linear mixed models with the response variables of total nitrogen, total phosphorus, Secchi depth, algae densities, and zooplankton densities, with fixed effects of gizzard shad abundance, so the number of shad captured per minute electrofishing, as well as the fixed effects of month, and then a random effect of impoundment. So first off, we'll look at our algae data. We were surprised that we found no significant relationship between gizzard shad abundances and total algal abundance. We also saw no increase in cyanobacterial abundance at higher abundances of gizzard shad, contrary to our predictions. So down here on the x-axis, we have log transformed shad abundance, and on our y, we have log transformed algal counts, and there are regression lines for each month. So we see no significant relationship between gizzard shad and either blue-green algae or the total algal abundance. Next up, we'll look at nutrients. So we found that total nitrogen and total phosphorus concentrations, tended to increase with gizzard shad abundance, but the relationship was pretty weak and dependent on month. So total nitrogen tended to increase at higher shad abundances in June and August, but not October. And for total phosphorus, there wasn't much of a trend in June or October, but total phosphorus did tend to be higher with higher shad abundances in the month of August. Next up is Secchi depth. So we did find that water clarity tended to be slightly reduced at higher shad densities, supporting our prediction. And this pattern tended to be stronger in June and August than it was in October. And then finally, we'll look at our zooplankton communities. We found evidence that zooplankton densities declined with increasing gizzard shad abundances. And we saw significant negative relationships between shad abundances and cladoceran and rotifer densities, as well as total zooplankton densities across taxonomic groups. And so this was a pretty strong effect, and you can see across all months there tended to be a decrease in zooplankton densities with increasing gizzard shad abundance, supporting our predictions. So to summarize, we observed no significant relationship between shad abundances and algal densities, and we did not see higher cyanobacterial counts in association with higher shad. Both the top-down and the bottom-up effects of shad should have stimulated algal communities, but we just didn't see that pattern. However, higher shad abundances were associated with lower zooplankton densities, higher total nitrogen and total phosphorus concentrations, and reduced water clarity. The relationships were not massive and exhibited a lot of variation, but were significant. And then overall, at high abundances, gizzard shad appear to have meaningful top-down effects on zooplankton communities. Shad were associated with increased nutrients and decreased zooplankton densities, but why didn't we see a change in algal abundance? Well, this could be for a variety of factors. So one of them is that could be a function of poor algal quantification on our end with our methods, or an insufficient sampling frequency, which made us miss some of the bloom events, made it more difficult for us to detect those changes. And then finally, these small impoundments exhibit a really high degree of spatial and temporal variability within each impoundment, making it more difficult to detect any effects of gizzard shad that might be present. These impoundments are also highly variable in terms of nutrient inputs and watershed land use, with the prevalence of agriculture being higher in certain parts of eastern and western Kansas compared to just central Kansas and then parts of western Kansas. And so in closing, advancing our understanding of gizzard shad ecology, as well as the ecology of other benthic feeding fishes in small impoundments, might help impoundment managers predict and prevent harmful cyanobacterial blooms in a time of unprecedented ecological change. And with that, I'd like to acknowledge the support of our collaborators with the Kansas Department of Wildlife and Parks, my dissertation committee. I'd like to thank the KSU Soil Testing Lab and the Kansas Department of Health and Environment. And finally, I'd like to acknowledge the assistance of all of the past and present members of the Giddo Lab for their help in the field and in the lab. And with that, I'd like to thank you for your time, for watching this talk, and feel free to hit me up on Twitter @lizardshad or email me with any questions or comments. Thanks, and have a great conference.